Okay, let's unpack this. Think about the sheer, mechanical violence of staying alive.
Yeah, it's pretty intense when you really look at it.
Right. Like every single cell in your body is constantly burning fuel, generating this toxic backlog of waste, and just demanding a relentless supply of oxygen.
And if that delivery system falters for even a few minutes, the entire biological engine shuts down.
Exactly. Yet we treat breathing like background noise. It just, happens. But today, we are taking a stack of A-level biology research to expose the literal mechanics of this invisible system.
It's a fascinating topic.
It really is. We are diving into the breathtaking engineering that organisms use to extract oxygen from completely hostile environments and force it into their cells.
Because while you might feel like you're just casually inhaling and exhaling, you are actually operating a highly complex biological machine. I mean, you are constantly manipulating physical pressure gradients.
Totally without thinking about it.
Right, without a second thought. You're exploiting microscopic fluid dynamics and relying on these shape-shifting proteins that actually read the chemical acidity of your blood in real time.
Which is wild.
It really is. If any one of those variables shifts out of balance, survival becomes impossible.
The crazy part to me is that this incredibly complex machinery only exists because of a fundamental math problem. Like, if we look at single-celled life, say, an amoeba in a drop of pond water,
Sure.
they don't bother with specialized lungs or gills. They just exist, and oxygen diffuses right through their outer membrane.
What's fascinating here is how strictly biology is governed by geometry. An amoeba works perfectly because it's microscopic, maybe 2 micrometers wide.
Really anything.
Exactly. So if you calculate its surface area compared to its internal volume, it has a surface area to volume ratio of roughly 3 to 1.
Meaning it has 3 times as much surface membrane as it has internal space.
You got it. Because it's tiny and its metabolic demand is incredibly low, oxygen can easily just diffuse straight out of the water across the membrane and instantly reach the center of the cell.
But the math breaks down the moment you try to scale up.
Oh, absolutely.
If that same cell simply grows from 2 micrometers to 4 micrometers, its volume increases exponentially faster than its surface area. The ratio plummets to like 1.5 to 1.
And as you move from single cells to complex multicellular animals, that ratio becomes a massive critical vulnerability. Your internal volume, the mass of living tissue demanding oxygen, becomes huge.
Right, while the surface area where gas exchange actually happens just can't keep pace. The diffusion distance from the outside air to a cell deep inside the organism becomes way too fast. I always visualize this as the ultimate potato problem.
The potato problem. I like that.
Yeah, think about if you try to roast a massive whole potato in the oven. The internal volume is huge, but the surface area exposed to the heat is minimal. It takes forever for the thermal energy to diffuse to the center.
Leaving the middle totally raw.
Exactly. But if you dice that potato up into tiny cubes, you've suddenly exposed a massive amount of new surface area to the ambient heat, and they roast in minutes. So how does nature solve this for highly active creatures?
Well, biology is essentially forced to create internal diced potatoes.
That is quite the image.
Right. Since large animals can't rely on their outer skin for gas exchange, evolutionary pressure resulted in specialized exchange surfaces. That means developing complex internal structures, like lungs in mammals or gills in fish.
Which basically fold massive amounts of surface membrane into a tight internal cavity.
Precisely. And crucially, it requires the evolution of a transport system, a fluid like blood, to bridge the gap between that exchange surface and the distant cells.
So if the goal is to pack as much surface area into the body as possible, biology takes completely different architectural paths depending on the environment. Take insects.
Yes, insects are brilliant.
I was genuinely surprised by the research here because insects completely bypass the use of blood for transporting oxygen.
They do. They utilize a completely different, highly direct mechanism. I mean, insects possess a tough exoskeleton made of chitin, which is practically impermeable to gases.
So air can't just pass through their skin.
Exactly. To overcome this, their bodies are lined with tiny pores called spiracles.
And those spiracles are equipped with muscular sphincters, allowing the insect to clamp them shut to prevent water loss, which makes sense, given that drying out is a constant threat at that size.
It's their biggest danger. When those spiracles do open, air enters a network of internal tubes called tracheae. These tubes are about 1 millimeter wide and are structurally reinforced with rigid rings of chitin.
To prevent them from collapsing under pressure, like a vacuum hose.
Very much like a vacuum hose.
Hmm.
The trachea then branch off into incredibly fine microscopic tubes called tracheals, which are a single micrometer or less in diameter.
Wow.
Yeah, these tracheals penetrate deep into the tissues running directly alongside and sometimes even into the individual respiring cells.
So it's literally an internal air pipeline. But this is where the fluid dynamics get so brilliant. If a bee is just resting on a flower, passive diffusion down those tiny tubes is enough.
Right, at rest, passive is fine.
But the moment that bee takes flight, its metabolic demand skyrockets. Passive diffusion physically cannot move oxygen fast enough. So how do they get more air?
The mechanism they use to actively pull air deeper into those microscopic tubes is such an elegant piece of chemistry.
Okay, lay it on me.
So the sealed ends of those tiny tracheals are naturally filled with fluid. During intense flight, the insect's muscle cells quickly exhaust their available oxygen and are forced to switch to anaerobic respiration.
Meaning respiration without oxygen.
Exactly. And a primary byproduct of this anaerobic pathway is lactic acid, which begins to accumulate inside the muscle cells.
Which instantly alters the cellular chemistry. The buildup of lactic acid lowers the water potential inside the cell.
That drop in water potential triggers osmosis. Water always moves from an area of higher potential to lower potential, so the fluid is physically drawn out of the tracheal tubes and into the muscle cells.
Wait, so the fluid level inside the microscopic tube drops?
Yes, and as it drops, it creates a subtle vacuum effect, physically pulling the column of air much deeper into the tracheal.
That is amazing. The very waste product of working hard chemically forces the plumbing to pull fresh air closer to the cells.
It simultaneously increases the surface area available for diffusion, too.
It's brilliant. But that trick relies on the physical properties of air. What about aquatic environments? Water is dense, highly viscous, and contains just a fraction of the oxygen found in the atmosphere.
Fish are facing a much steeper uphill battle, for sure. A highly active bony fish, like a tuna, has a massive oxygen requirement, but it's surrounded by a medium that actively resists giving up what little oxygen it holds.
So how do they manage?
They solve this through a unidirectional flow system. Water is drawn into the buccal cavity, the mouth, and forced out through the operculum, the protective flap on the side of the head.
But the actual extraction happens as the water washes over the gills.
Right.
The structural design of the gills is entirely focused on surface area and short diffusion distances. You have the gill arches supporting numerous filaments, and those filaments are covered in tiny folded structures called lamellae, which are packed with capillary beds.
But the real engineering marvel here is the countercurrent exchange system. If we connect this to the bigger picture, this system represents one of the most perfectly optimized physical processes in nature.
That's a big claim.
It earns it. Inside those tiny gill lamellae, the blood within the capillaries flows in the exact opposite direction to the current of water washing over the outside.
Okay, I actually want to push back on the efficiency of this for a moment. Yeah, because at first glance, parallel flow, where blood and water move in the same direction, seems like it would result in a faster uptake. Like, if highly oxygenated water hits completely deoxygenated blood simultaneously, you have an incredibly steep concentration gradient. Wouldn't that massive initial diffusion spike be preferable?
I see where you're coming from. It does create a rapid spike, but it is fundamentally wasteful over the length of the capillary.
How so?
Well, if blood and water flowed together in parallel, that steep gradient would cause rapid diffusion initially, but they would quickly reach equilibrium. The water would drop to 50% oxygen saturation, the blood would rise to 50%.
And then the concentration gradient is eliminated.
Exactly. Diffusion would permanently stall. The fish would leave half the available oxygen behind in the water.
Oh, wow. But by running them countercurrent against each other, the blood arriving at the lamellae has almost zero oxygen, and it encounters water that has already been mostly depleted by its journey across the gill.
Yet crucially, that nearly depleted water still holds slightly more oxygen than the newly arrived blood. So diffusion occurs.
I get it. And as that blood flows forward, gathering more and more oxygen, it is constantly swimming upstream against increasingly oxygen-rich water.
Right. Even as the blood reaches 70 or 75% saturation, it encounters fresh, incoming water at 100% saturation.
So the concentration gradient is permanently maintained across the entire anatomical length of the gill. Equilibrium is completely avoided.
Allowing the fish to extract up to 80% of the ambient oxygen. It is an absolute masterclass in gradient manipulation.
Mammals, on the other hand, don't use countercurrent exchange. We are warm-blooded. Maintaining a constant internal body temperature requires an astronomical metabolic rate.
It's a huge energy cost.
And we pull dry, often freezing air into a delicate internal environment.
Because of that, the mammalian airway is far more than just a delivery pipe. It is a heavy-duty conditioning and filtration system. When air is drawn through the nasal cavity, it's warm to body temperature and highly moisturized.
Then it travels down the trachea, splits into the bronchi, and branches out into the smaller bronchioles.
And the walls of that airway are constantly working. The trachea and bronchi are lined with goblet cells pumping out sticky mucus to trap inhaled dust and airborne pathogens.
Alongside them are ciliated epithelial cells, waving these microscopic hair-like structures in a synchronized rhythm to sweep that contaminated mucus up toward the throat.
Where it can be swallowed and destroyed by stomach acid, it's an autonomous, self-cleaning escalator.
And to keep this critical pathway from collapsing under the negative pressure of breathing, the trachea is structurally reinforced with flexible cartilage.
Exactly.
Looking at the structural diagrams, though, it highlights that these cartilage rings are actually C-shaped. It seems like an architectural compromise to leave the posterior side of the windpipe completely unshielded. Why not a fully enclosed structural ring?
It is absolutely an anatomical compromise. You have to consider what shares that space. The esophagus, the muscular tube transporting food to the stomach, runs immediately posterior to the trachea.
Oh, right behind it.
Right. If the trachea were encased in rigid, complete circles of cartilage, every time you swallowed a dense bolus of food, it would strike the back of the windpipe and jam.
Ouch.
Yeah, not ideal. The open part of the C faces the esophagus, allowing the muscular tube to temporarily distend into the tracheal space during a swallow without causing an obstruction.
So form heavily follows function. All of this architectural routing eventually dead ends at the alveoli, the actual site of gas exchange. We are looking at hundreds of millions of microscopic air sacs.
Providing a surface area roughly the size of a tennis court packed into your chest.
A tennis court? That's absurd. And the walls of these alveoli are formed from squamous epithelial cells that are just a single cell thick.
And the surrounding capillary networks are equally thin. Red blood cells practically have to squeeze through in single file.
Pressing themselves right against the capillary wall.
Exactly, which reduces the diffusion distance to an absolute biological minimum. Additionally, the interior of these alveoli is coated in a vital fluid containing pulmonary surfactant.
The surfactant is fascinating because it serves a dual purpose. Gases must be dissolved in fluid before they can diffuse across a membrane, so the moisture is completely necessary.
Yes, but the surface tension of water naturally wants to contract.
Right, so without surfactant, that tension would pull the delicate alveoli inward, causing the lungs to completely collapse after every breath.
The surfactant breaks that surface tension, keeping the microscopic sacs inflated.
But having a massive surface area of perfectly inflated alveoli means nothing if we can't constantly refresh the stale air inside them. We have to maintain the oxygen gradient.
This is where we rely on the physics of ventilation, heavily dictated by Boyle's law. Boyle's law establishes that the pressure of a gas is inversely proportional to its volume, provided the temperature is constant.
Meaning if you force a gas into a larger volume, its internal pressure drops.
You got it.
The classic physical demonstration of this is a standard medical syringe. Like if you cap an empty syringe with your thumb to seal it and pull back on the plunger, you feel the suction pulling your thumb inward.
The perfect analogy.
By physically expanding the volume of the plastic barrel, you drop the internal pressure below the ambient atmospheric pressure in the room. The atmosphere immediately wants to rush in to equalize it.
And that physical principle is what powers inhalation. Breathing in is an active muscular process. Your diaphragm, which normally rests in an upward dome beneath the lungs, contracts and flattens downward.
While simultaneously, the external intercostal muscles located between your ribs contract, pulling the entire ribcage upward and outward.
Expanding the thoracic cavity, dropping the internal pressure, and allowing atmospheric pressure to simply push air down the trachea to fill the void.
Exactly the same as pulling the syringe plunger.
Now, during normal resting ventilation, exhalation requires almost no muscular effort. It's passive.
You just start flexing.
Pretty much. You simply stop contracting those muscles. The diaphragm relaxes back into its dome shape, the weight of the ribcage settles inward, and the natural elastic fibers woven around the alveoli recoil back to their resting state. The thoracic volume shrinks, internal pressure spikes, and the air is expelled.
But if you transition from a resting state to an all-out sprint, exhalation shifts from a passive recoil to a highly active muscular drive, right?
Oh, absolutely. Forced exhalation recruits a whole new set of muscles. The internal intercostal muscles contract, actively hauling the ribs downward and inward.
Concurrently, the abdominal muscles contract, forcefully shoving the internal organs upward against the diaphragm.
These muscles work antagonistically, in direct opposition to the inhalation muscles, rapidly collapsing the thoracic volume to clear the lungs much faster.
So Boyle's law handles the gross movement of air into the thoracic cavity, and the massive surface area of the alveoli facilitates the diffusion into the blood. But once oxygen crosses that squamous epithelium, we hit a massive transport bottleneck.
We do.
Oxygen dissolves incredibly poorly in aqueous plasma. If we relied on free-floating oxygen, our blood would carry almost nothing.
Which is why we rely on a dedicated chemical courier, hemoglobin.
The superhero of the blood.
Hemoglobin is an extraordinary, complex protein housed inside your red blood cells. It possesses a quaternary structure, meaning it's assembled from 4 distinct polypeptide chains.
Embedded within each of these 4 chains is a heme group containing an iron ion.
And it is this specific iron ion that binds to the oxygen. Therefore, a single hemoglobin molecule can carry exactly 4 molecules of oxygen.
The mechanics of how this protein loads and unloads oxygen is where the chemistry really shines. If you plot hemoglobin's affinity for oxygen on a graph, the oxygen dissociation curve, it doesn't trace a straight linear path.
No, it plots an S shape.
Due to a phenomenon known as cooperative binding. I like to picture this using a rideshare analogy. Imagine a 4-passenger car where the very first door is rusted shut and incredibly stubborn.
Okay, I'm with you.
The first passenger has to struggle violently to yank it open, but the moment that first door opens, it triggers a mechanical release, and the other 3 doors instantly pop wide open for the remaining passengers.
That analogy captures the allosteric nature of the protein perfectly. When hemoglobin enters an environment with a low partial pressure of oxygen, its molecular shape is tight, it has a very low affinity and resists binding.
Like the rusted door.
Exactly. But in the lungs, where oxygen pressure is immensely high, that first oxygen molecule forces its way onto a heme group. The moment it binds, it causes an allosteric shift, a physical change in the 3D shape of the entire quaternary protein structure.
Which opens the other doors.
Yes. This specific shape change dramatically alters the remaining 3 heme groups, making it exponentially easier for the second, third, and fourth oxygen molecules to bind.
But loading the oxygen into the blood is only the first logistical hurdle. The far more complex problem is targeted delivery. How does the hemoglobin know where to release the oxygen? Flooding resting tissue with oxygen is a waste, but a sprinting leg muscle needs a massive immediate drop-off.
If we connect this to the bigger picture, this is the sheer elegance of the Bohr effect. The system utilizes the cellular waste product itself as the primary chemical signal.
Because actively respiring tissues produce high quantities of carbon dioxide.
Right, and when that carbon dioxide diffuses into the red blood cell, it doesn't just sit there. It reacts with the water inside the cell, a reaction rapidly catalyzed by the enzyme carbonic anhydrase, forming carbonic acid.
And acids dissociate. The carbonic acid breaks down into hydrogen carbonate ions and free hydrogen ions.
And these free hydrogen ions immediately threaten to drop the pH of the blood, making it dangerously acidic. To buffer this, the hemoglobin protein itself binds to the hydrogen ions.
But binding those hydrogen ions triggers yet another allosteric shift.
Precisely. Just as binding oxygen changed the protein's shape to increase affinity, binding hydrogen alters the shape to aggressively lower its affinity for oxygen.
Wow.
The hemoglobin physically cannot hold onto the oxygen molecules anymore, and it ejects them.
It is such an elegant feedback loop. The more intensely a muscle works, the more CO2 it generates. And that localized spike in acidity chemically forces the passing blood to drop its oxygen payload exactly at the site of highest demand.
It shifts the entire S-shaped dissociation curve to the right.
It does. And a detail often overlooked is the chloride shift. As the negatively charged hydrogen carbonate ions diffuse out of the red blood cell into the plasma, negatively charged chloride ions rush into the cell to preserve the delicate electrical balance across the membrane.
It's a flawless chemical balancing act, and nature heavily customizes this S-curve for specialized environments too.
Oh, like fetal hemoglobin.
Yes, fetal hemoglobin, for instance, has a dissociation curve shifted significantly to the left compared to an adult. It operates with a naturally higher affinity for oxygen, allowing the fetal blood to literally pull oxygen away from the mother's hemoglobin across the placental barrier.
That makes total sense. We see the same leftward shift in animals adapted to high altitudes, like llamas, allowing them to snatch up oxygen in incredibly thin atmospheric conditions.
It's all about tuning the chemical affinity of the protein to match the environmental or developmental reality of the organism.
We've examined the microscopic fluid dynamics, the structural compromises, and the allosteric shapeshifting. But practically speaking, measuring these precise metrics in a living, breathing human requires some clever lab work.
You certainly can't visually track individual oxygen molecules moving through a patient's bloodstream.
No. To quantify this, researchers rely on a device called a float spirometer.
It's an older but remarkably ingenious mechanical setup. The subject breathes entirely through their mouth into a tube connected to a large, sealed plastic chamber that floats upside down in a tank of water.
So as you inhale, you draw air out of the floating chamber, causing it to sink into the water. When you exhale, you push air back in, causing the chamber to rise.
And a pen attached to the chamber draws a continuous trace on a rotating drum of paper. By reading the amplitude of those waves, you can easily find the tidal volume, the amount of air moved in a normal resting breath, and the breathing rate.
You can also measure the vital capacity by having the subject take a maximum inhalation followed by a maximum exhalation.
But the most vital piece of data comes from observing the trace over a period of minutes. As the subject breathes normally, the entire wave pattern slowly angles downward.
Meaning the total volume of gas inside the sealed system is steadily decreasing.
Yes.
This is the part of the setup that I found genuinely brilliant as a proxy measurement. If you just breathed into a perfectly sealed tub, the volume of gas wouldn't change at all because for every molecule of oxygen your cells consume in aerobic respiration, they produce roughly 1 molecule of carbon dioxide in return.
Right, the trace on the graph would just bounce horizontally forever.
Which brings us to the most critical component inside the spirometer, soda lime.
Yes, soda lime. By placing a canister of soda lime inside the circuit, you actively strip the exhaled carbon dioxide out of the gas mixture.
It absorbs it completely.
This raises an important question about how we observe biology. We often can't look directly at the cellular engine, so we measure the exhaust. Because the soda lime instantly removes the exhaled CO2, the physical loss of volume in the floating tub becomes a direct, real-time measurement of the oxygen that has been permanently locked into the patient's metabolism.
So measuring the downward slope of that graph calculates the exact rate of cellular oxygen uptake without ever drawing a drop of blood.
It's beautifully simple.
It really is. So when you synthesize all of this, what does it mean? It means every single time you take a breath, you are executing an unbroken chain of phenomenal physics and chemistry.
An incredible chain.
You are utilizing Boyle's law to manipulate atmospheric pressure. You are solving complex surface area to volume math with hundreds of millions of microscopic surfactant-coated sacs. And you are relying on allosteric iron proteins that read the microscopic acidic exhaust of your tissues to deliver fuel with absolute precision.
It is a continuous high-stakes relay race from the atmosphere in the room all the way down to the mitochondria inside your cells.
Which leaves you with an incredible final thought. We know that biology already tweaks the system. Fetuses and llamas possess hemoglobin naturally tuned to grab oxygen in scarce environments.
True.
Could humanity eventually use targeted gene editing to rewrite our own hemoglobin dissociation curve?
Oh, wow.
Right. Could we intentionally shift our affinities so far to the left that humans could free dive for hours in the crushing depth of the ocean, or survive in the thin alien atmosphere of another planet without bulky life support suits?
That is a wild concept.
The biological mechanics already exist in nature. It's simply a matter of how we might eventually rewrite the code. Something to think about the next time you take a breath.